Okay. Hi, everyone. My name is Margie. I am one of the program coordinators for the Innovation and Design Hub. I have Cynthia right here, right next to me. And I also would like to introduce everyone to Ana Moreno. She is the CEO and co-founder of Navega Therapeutics, a preclinical stage company focused on developing next generation epigenetic gene therapies and pain for neurological disorders. Those are really big words. I'll
Great.
give you the stage now.
Thank you so much, Margie and Cynthia, for the invite. Hi, everyone. Like they mentioned, my name is Ana Moreno. Very excited to be here today and kind of just share with you some of my journey. Also, I have prepared some slides today that are exactly the slides that I present to investors. So just to give you an idea of what kind of presentation would want to give to investors. And so with that, let me go ahead and share my screen. So as Margie mentioned, at Navega, we are focused on developing fully human epigenetic modulators for gene and cell therapeutics. And I'll explain what these aspects mean as I continue with the conversation. But before that, I wanted to give a little bit of the timeline or my journey of how I started the company. So just a quick background. I was born in Mexico. I moved to the US during high school in Yuma, Arizona. After which I went to undergrad in Tucson at the University of Arizona. And there I studied both French literature as well as biosystems engineering. I really just wanted to have that kind of balance between science and the more humanities. After that, I really wanted to be at the highest level that I could be in my specific area of focus. And so I decided to pursue a PhD at University of California, San Diego, so UCSD here in San Diego. And to be honest, it was always tough to know what kind of research do I want to go after, what do I want to do with my career. I didn't really know at that time point. It wasn't until I joined my lab in Prashant Mali's lab in bioengineering, where I was working on gene therapy. And for me, that's always been a very exciting space. Like, can we actually develop gene therapies to help patients, but not just work in the lab and publish, but can I actually push this ahead into the clinic? And so that was kind of my mindset back then. I started the PhD in 2015, and I was working with CRISPR. So CRISPR, as some of you might know, it's a bacterial protein that works to cut DNA, and we have repurposed it now to actually help with gene therapies in the human setting. So I was working with a variant of CRISPR called dead-Cas9, where we were not editing the genome, but we could turn genes on or turn genes off. After I learned about a mutation in the human genome in the sodium channel called NaV1.7, in which people feel no pain whatsoever, I started thinking about the opioid crisis in the US, where there's a lot of people that are addicted to opioids due to the amount of pain that they're in. And so I told my advisor, what if we take this platform using this dead-Cas9 and apply it to this gene and see if we can have great results in mice? And so that's how this all got started back in grad school. When we started seeing some really great data, that's when we decided to incorporate the company. So you can see in the timeline, I was still a PhD student when we started Navega Therapeutics. I decided to take business classes. So take advantage, I guess, one of the feedback or thoughts I would have is universities a lot of times have great programs that can help you out. And so UCSD had a class called Lab to Market, where they incorporated PhD students with business students. And that really exposed me to the business world. And I think it doesn't matter what kind of career you decide to go after. It's always great to take some business class because we all really need it, even into your day-to-day financing, but also when you're thinking about vision of a company. So this really helped me to hone in on how can I communicate the science to more of the business community. In 2019, I graduated in April with my PhD. But at the same time, while I was a grad student, I decided to start applying for non-dilutive funding, which means grants. So grants to really fund my startup, my company. And so I received with my co-founder 2 SBIR grants that were around $1 million. And so that really enabled me to start my company. And we moved into a space called J Labs. So Janssen Pharmaceuticals has an incubator space where there's a lot of startups, actually 60 startups here. And you pretty much sublease a lab space for your company, because it's obviously very expensive to go after really large space. So this allows you to have your small team, small space, but also there's shared equipment. So you don't have to have that really expensive upfront cost. So, we moved in right after there's a pandemic that started. So you can imagine in the first few months of the pandemic, a lot of us were working from home. So we decided to continue working with grants, continue doing research at home. And since then, we've gotten some proof of concept in vivo in mice, and now we're actually $8 million in grants. And then, this last year, something that I've learned is learning what investors are looking for. And one thing I've learned in that sense is that a lot of them like to invest in something called a platform, meaning you have a technology that can be applied to many different aspects, many different diseases. And so at this time point, we decided to merge a different company into Navega. They're called TBG. And they were bringing a really exciting platform that I'll talk about today. And so this really enabled us to go about our fundraising for something called a Series A. So in the lifetime of the company, usually you have a seed round, a Series A, a Series B, and those have different, different sizes of funding. The seed round, for example, can be anywhere from 1 million to 10 million. The Series A, I've seen 10 million, I've seen 100 million, and so on. So this really is to push forward the company and fund it for the next 2 to 3 years. I'm really happy to say that we've signed a term sheet with a lead investor, meaning that there's an investor that wants to lead our Series A. And so in the next few years, we're going to focus on developing the platform and then filing with the FDA with our drug in around 2025. So at this time point, I'm going to start shifting into the deck that I use with investors. So this is the rest of the founding team. The original 3 founders were Fernando, Prashant, and myself. Fernando has a Ph.D. and PharmD from Spain, and he came to the US to do postdoc studies. And we've met through a collaboration. So he was at Scripps and I was at UCSD, and we were collaborating on a method of how to evade immune responses that gene therapies might trigger. Prashant was my PI, so he was my advisor in grad school. So he was one of the first to demonstrate that CRISPR works in mammalian cells. And he's also very entrepreneurial. So he has 2 other companies. One's called Shape, one is called Boundless. And so what really also enabled me to start my company is that he's also very open-minded and open to innovation, open to patenting everything. And so that really helped out in terms of starting the company from there. These 3 other members here, there were the 3 that came into the company when we merged. So Marcus Noyes, he's a zinc finger expert, and I'll explain what zinc fingers are in a little bit. Miko is a functional genomics expert, and then Philip is a computationalist as well as an AI expert. So I'll go about and explain the platform, and you'll understand why we brought these people in the company as well. So our value proposition is that we built our first in-class zinc finger platform. So zinc fingers are DNA binding domains. So these are proteins that naturally exist in the human body. And they typically work with transcription factors to turn genes up and turn genes down. But as scientists, what we've done is we come to a place where we understand these proteins really well, so we can design our own. And that way we can design where we want to go in the genome. Which gene do we want to go after? So for example, if you're looking at Parkinson's, there's alpha-synuclein is a gene of interest. For Alzheimer's, there's tau. It's a target that you can go after. So really designing zinc fingers to go after these really exciting, and complex diseases. In addition, we've actually built an AI model in which, because proteins are difficult to design, we can design proteins really quickly. We insert the sequence where we want to target, and that specific model gives us the sequence that we need to use for our therapeutics. We have also the ability to reprogram human transcription factors with designed zinc fingers, as well as a proprietary set of fully human activation and repression domains. In vivo proof of concept for gene therapy and cellular reprogramming targets. And as I mentioned, $8 million in undiluted funding raised, so grants. We have licensed IP from the universities. We are set up in San Diego, and soon, actually, Marcus, one of the co-founders, will be leaving his job as professor at NYU and joining full-time as CTO. So we're going to have a site in New York as well. And so what kind of diseases do we focus on, and why do we care? So we're focused on misregulation. And what does that mean is that when someone has a specific mutation in a gene where that gene actually is expressed too much or expressed too little, we want to go and actually go and fix that gene so that it goes back to what it should normally be doing. And in the past few years, there have been a lot of studies in which for very complex and common diseases, such as in cardiovascular, neurological, diabetes, we're seeing a lot of misregulation of genes. And so as a company, we're focused on developing therapies to go after this misregulation. Some challenges are delivery. So we use something called adeno-associated viruses, so AAVs. These are viruses that we can use to send something into a cell. However, CRISPR is really large, so that's one of the challenges. The other challenge is immunogenicity. So again, Cas9 comes from a bacterial protein. So these are the original thoughts of the platforms that are used by some of our competitors. So how are we solving some of these issues is that we're first using fully human proteins. So zinc fingers actually exist in more than 50% of transcription factors in the human body. So the body already recognizes them. That's a huge advantage. Second advantage is that they're really small. So you can deliver multiple of these in one single virus. And as I mentioned, the fact that they're fully human really reduces the risk of immunogenicity. So Navega mission, really, we're excited about tackling common disease with epigenome regulation. Using zinc fingers, applications in gene therapy will be focused on in vivo single and multiplex gene regulation. So turning one gene or more than one gene up or down. And cell therapy, we're going to be reprogramming cells inside the body. And I'll give some examples of both of these as I continue with the conversation. But very importantly, something I learned when I started being an entrepreneur is, as a scientist, we think that everyone really wants to work with our therapies, is very excited about what we're doing. But the FDA, at least for gene therapies, they're more comfortable with rare diseases in gene therapy, mainly because gene therapies are complex products. And so how are we going to focus on entering the market? Here, we're going to focus on common disease that's also rare. So I'll give an example of a rare disease that is an example of a common disease. So we'll first start with rare, and that same drug product can then be utilized for common disease. So I'll explain a little bit on the platform, dive into some applications, and then get into some corporate development. So first here, focus on how do we reprogram human transcription factors. And so first, explaining what is epigenome, what does that even mean, right? So when you look at the central dogma of biology, DNA is transcribed into RNA, which is then translated into protein. And most of the drug products that you see that are FDA approved are the ones that you take or someone else might take, either target the RNA or they target the protein. However, there's some issues with these targets. One is that there's high turnover, meaning that RNA molecules change with time, they increase or decrease. And the issue with targeting the protein is that you can have a lot of lack of specificity. So a small molecule might bind to many different areas in the body, and that will cause issues. So what we're doing is we're turning genes on or off, and that's really the epigenome approach, where we're not creating any permanent changes in the genome. We're just turning things up or down. And this is what I mentioned earlier, we're using zinc fingers. So these are a cartoon of zinc fingers, and each zinc finger will recognize 3 base pairs in the human genome. And to create a zinc finger array, or like a link of multiple zinc fingers that will be very specific, you need 6 fingers to engage with the DNA. If any subset of these zinc fingers attach to the genome, you can have tens of thousands of off-target effects or of other genes that are being modulated. So what one of our founders, Marcus, has been working on is specifically understanding protein-protein interactions. So when proteins are right next to each other, they'll have an effect on each other, and that will have an issue on the specificity. So what he's done is trying to understand, when zinc fingers are right next to each other, how can we work and ensure that they are compatible? And the way he's done this is using a system called the bacterial hybrid system. So what does this mean? You first have a library. So these are many different zinc fingers that will bind to, in this example, CAC. And right next to it is a zinc finger that we know exactly will bind to AAG. We know how it works specifically. And so whenever a zinc finger will attach to CAC, this will turn on a gene called His3, and that will allow the bacteria to survive. If this is not turned on, the bacteria will die. And so the bacteria that survive, we can then sequence, and we'll know exactly which zinc fingers will bind to that sequence. And he's done this for 23 different libraries. That's 94 billion protein DNA interactions. So really understanding when the whole genome, which zinc fingers work well and how do they actually interact with each other to make sure that we have compatible zinc fingers. All this information has been fed into an AI model. And the reason being is that zinc fingers are actually complex to design. When you think about CRISPR has something called a guide RNA. It's really much just an RNA sequence, and a lot of people can work with it. But zinc fingers are a lot more complex. And so this enabled us to design zinc fingers just as quickly as you would design a guide RNA. And Marcus has over 20 years of experience in this space, and since he's joined the company, we can also go about and achieve therapeutic grade specificity. Something that's also very unique to the company is the ability to reprogram human transcription factors. So here's an example of a transcription factor called KLF6, and these use zinc fingers. So in nature, this will bind to the DNA of interest, a sequence of interest, and this will increase the expression of the gene of interest. What we've done is we replaced the native zinc finger with our own design zinc finger, where we tell it, where do you want it to go in the human body? And why is this exciting is because we can tell it to go after genes that cause disease in the body. So as an example, what we do is we canvas a region of the gene that we want to change the expression. We do something called quantitative PCR, where we can see if that gene is being upregulated. And as you can see, a few of the examples indeed are being upregulated or expression is increased. And lastly, we looked at genome-wide RNA sequencing. What does that mean? This tells us if any other gene in the genome is being changed with our therapy. And you can see that only the single gene, the gene that we care about, is being changed. So this is very exciting. Here we are showing that we have activation domains that are fully human. So again, you always want to differentiate yourself with what others are doing. And this is one example of how we're different. Everyone in the industry is using something called BPR. And BPR is a way to activate genes, but it has proteins that are actually viral in origin. So one of our founders has screened a lot of different proteins and found a few of them that are fully human, where you can activate a gene of interest with a human protein. And again, we care about this because of the immune response. We've also done this for repression. So repression means to turn down the expression of a gene. And again, everyone in industry is using something called a COX-1 crab. These are naturally occurring in the body and they can repress expression of a gene. But our founder found a few of them that are very strong. And why do we care? Is that some diseases require you to have very low expression to actually help the patient. And also this could potentially allow you to lower the dose of your drug. And this is an example of how we can repress genes. So specifically, as I mentioned in the last slide, we found a specific crab domain, a ZIM3 transcription factor that represses genes of interest. Again, we replaced the natural zinc finger with the one that we designed to target a gene of interest. And then we look at quantitative PCR. Here you see a no zinc finger control. So this is the natural state of that gene. You can see that all the zinc fingers that we designed work as compared to control, but a few of them are very strong. So these two give us over 90% repression of that gene. And so these would, for example, be great targets to move forward into the clinic. So then what is the strategy of the company? Obviously, when you have a platform, that means you can do a lot of things, but as a company, you want to be focused. You want to focus on a few things. And so the strategy when we're thinking about moving ahead is we're going to be partnering with large pharmaceutical companies on more complex diseases, such as in pain or neurodegeneration. However, for ophthalmology, that will be focused mainly into the company. The reason is that ophthalmology is a small space. It's very already well understood, and there's already FDA-approved drugs, gene therapy in the eye, whereas the brain is complex. Even delivery into the brain is very difficult with gene therapy. So that's why difficulty, more risky targets we can partner out, and then less risky, more straightforward, we can keep in that company. But the ultimate dream of the company, the ultimate goal is the ability to reprogram cells inside the body. So typically, a lot of times people might require a transplant of a tissue. They might require, cells that are engineered outside of the body and then are delivered in the body. In that process, a lot of these cells die, and a lot of times it's not efficacious. So our goal, for example, is to reprogram glial cells to neurons in the brain to help a lot of people that have had Alzheimer's or Parkinson's. In the eye, we already have done some work where we can reprogram a specific cell type to another to help with visual acuity. Or if someone has an infarction, for example, we can reprogram potentially into cardiomyocytes. So I'm going to give you some examples of gene therapies that we've done. The first, we're focused on pain. So this is what I mentioned that I did in grad school, where there's a loss of function mutation in a gene called SCN9A. This gene encodes for a sodium channel. So this channel is involved in the transmission of pain. And so this mutation causes a decreased function of NaV1.7, in which people then feel no pain whatsoever. So they feel nothing. Obviously, that's not a good thing. You want pain is important for the body. It's important to understand when something is not going well. However, this gene gives us an insight into a target that we can go after because people that have a mutation in this gene don't really have any other side effects. There's also another population in the humans that have a gain of function mutation in this specific gene. That means that there's an increased function of NaV1.7 ion channels. How do you imagine an ion channel? It's kind of like electricity. Imagine if you touch something with your finger that's hot, that sends a signal that goes all the way into your arm, up your back, and into your brain. So if the channel is overactive, it means it's opened and that electricity is actually happening a lot quicker and more often. That's what causes the pain. And these patients, you can see in the picture, they have pain in their extremities, their hands, their feet, their face. And how they usually feel better is that they actually place their extremities into cold water or use fans to help with the pain. But obviously, this has issues because it can cause ulcerations and hospitalizations. So, how are we going to move this therapy forward into the clinic? When I mentioned earlier that we're first focused on rare disease, and then that same drug product can be utilized for other common pain. Here, we're focused on primary fibromyalgia to show proof of concept first, and then in the phase 3 clinical trial, because there's phase 1, 2, and then 3. There, we can focus on a larger pain population, such as people that have low back pain, for example, or that have pain after receiving chemotherapy. And so we have proof of concept in vivo where we deliver a zinc finger with a repressor. So here, as I mentioned, we want to downregulate the expression of NaV1.7 to help with pain. This is how we deliver it. As I mentioned earlier, we use viruses, adeno-associated viruses to deliver. So these viruses, it pretty much is like transportation. So we take out all the viral proteins and it's just the capsid. It's just the outside that we use. And we put in there a plasmid vector that expresses the zinc finger and repressor. So this is placed into the virus. The reason we use viruses here is because they don't really have a strong immune response. They're non-pathogenic. They also are really smart. So viruses have obviously in the evolution have learned how to get into cell types. They know how to get into neurons. They know how to get into different cell types. So we need that kind of delivery system to get our gene therapy inside the body. And the route of administration for this is an intrathecal injection. So these are similar to an epidural, to what a woman will receive when she's going through labor. They go a little deeper into the cerebrospinal fluid. And the reason we do these injections is because we're targeting a cell type called dorsal root ganglia. These are, as you can see, these are little round cell bundles that are located up and down your spinal cord. And that's where all the nerves connect and the cell body of NaV1.7 is located before going into your brain. So if you can stop the pain signal at the back level, then you can actually stop people from feeling pain. So we tested this in vivo in a mouse model. Specifically, this is a mouse model of chemotherapy-induced pain. So chemotherapy, when a patient has a chemotherapy treatment, a lot of them actually develop both mechanical pain and cold pain. That means pain like grabbing things, just in extremities, pain in their fingers and their feet, or grabbing things from the fridge or cold weather. And what's really interesting is that chemotherapy has the same effect in mice as well as in non-human primates. So for this model, we either have a group of mice that receive no paclitaxel. So these mice are not in pain, they're just naive. One group that received gabapentin, a small molecule at a high dose, an AAV zinc finger targeting NaV1.7, or an AAV M-Sherry negative control. On the y-axis, we're looking at withdrawal threshold in grams, which is we apply these filaments called biofibre filaments onto the hind paw of the mice. They're on a platform. And then the mice respond by licking or flicking their paw, meaning they feel pain. As you increase in the y-axis, they feel less pain. As you decrease, they feel more pain. So at day 0, we perform something called a baseline. It's just having a baseline before any treatment. As expected, there's no significant change between the groups of mice. After the chemotherapy, we can confirm that the 3 groups of mice that received the chemotherapeutic indeed had this mechanical pain. And so after the gene therapy, we were excited to see a nice reversal in this mechanical pain with the same finger targeting NaV1.7. So you can see that we were able to make the mice feel no pain again. And that was more significant than the gabapentin small molecule. So gabapentin is a drug that's over the counter that's used for pain. And so we can show that it's more efficacious with our gene therapy. And the negative control, the M-Sherry, which is a fluorescent marker, you can see that there's no change over time as expected. So this work has been published in Science Translational Medicine, and we have shown that it works in multiple animal models of pain, including inflammation, as well as central pain. It's long lasting, so one injection provides more than 10 months of pain relief, highly specific. So other pharma has tried to go after this gene. However, there's been lack of specificity because there's a different isoforms or different kinds of sodium channels in the body. One of them is a sodium channel called NaV1.5. And so a lot of times a small molecule will bind to NaV1.5 in the heart. And you can imagine you don't want cardiac issues. But we've shown that we can go after one single channel. We've done a lot of safety studies showing that there's no motor, olfactory, or sensory side effects. As I mentioned earlier, it's applicable across pain conditions. This program has received a lot of money in non-dilutive funding, and we're really excited about that because there's a huge opioid epidemic in the US, and there's a lot of support from the NIH for this kind of work. And like I mentioned earlier, we aim to partner this program as we're focused on partnering neurological disorders. So now I want to focus on something that I think is really unique and exciting, and that's cellular reprogramming. What is that? That means that you can convert one type of cell to a different type of cell that has a different function. And we can do this in vivo. And here we're focused on a disease called retinitis pigmentosa. This is a retinal dystrophy that affects 1 in 4,000 people. It causes progressive degeneration of a cell type called rods. These are the cell type in your eye that has night vision. After that dies, then your other cells die, so the ones that are called cones, and those have the day vision. And so you can see a normal retina, you have a nice architectural structure, your rods, your cones, the outer nuclear layer. But people that have retinitis pigmentosa, you can see that the architecture is messed up, and this causes blindness. So obviously it's a big issue, but the challenge is that there's over 200 underlying mutations across 50 genes. What does this mean? Is that you would have to design multiple gene therapy products to go after each of these mutations. That require billions of dollars and a lot of different companies to actually work on this. So our approach is something called a mutation agnostic approach, where we're not going after one mutation. And the way we do it is by reprogramming rods to cones. So retinitis pigmentosa, obviously, when you have a mutation in your body, it's in every single cell type, but it's been shown to only affect rods and not cones. So if we can reprogram rods to cones, then we can prolong visual acuity because cones are not affected. And we do this by targeting a mouse model. These mice are called RD10 mice. These mice are a mouse model of retinitis pigmentosa, and they become blind by day 60, meaning after 60 days, they can no longer see. So we stain for rods, the night vision, the cones, and the outer nuclear layer. On the bottom, we delivered a dead Cas9 system to repress NRL. So this means that we're downregulating the expression of NRL. And that gene is really interesting. This gene is a master regulator of rod versus cone. So if you can turn it off, that means that the cell type will reprogram from rods to cones. And that's exactly what we've shown. You can see that there's an increase in cone photoreceptors by repressing of one gene, this NRL gene. In addition, whereas you see sparse nuclei here in the architecture of the eye, we see 2 to 3 additional layers in the eye by reprogramming and repressing that single gene. And the most important experiment is, can the mice see better, right? Can they see better after this gene therapy? And indeed, you can see between the control and our therapy, we saw a nice increase in the visual acuity in the mice. So we're really excited about this program, and we are currently designing zinc fingers to target NRL as we move this forward into the clinic. So now I'll talk a little bit about corporate development. We are currently raising a 50 million Series A. As I mentioned, companies usually start with a seed round, Series A, and whatnot. So why do we need this much money, right? It's a lot of money. So what are we going to do with it? The first is operations, expanding the team. So we're currently 6 people, 6 full-time employees. With this 50 million, we're going to grow to 40. There's going to be 30 in San Diego, and San Diego is going to be focused on translation. So working on all the diseases that we care about, as well as manufacturing. And then the site in New York is going to be focused on continuing to develop the platform and make it better. The first few months, we're going to be focused on disease and target selection. So working closely with our investors to pick which indications to go after. It's a complex process because, not everything has pros and cons. Maybe the biology is not well understood. Maybe the clinical design is very difficult. Maybe there's a lot of competition for that specific gene. So it's really about understanding what's the strategy of what to move forward. So in terms of the NaV1.7 program, we're going to be pushing that along with a lot of grant funding. The next steps for us are to choose a lead candidate. That means the gene therapy that's going to be delivered into humans. After that, it's manufacturing. So CMC is manufacturing. And manufacturing is a really long timeline because it requires a lot of virus to actually put into the patient and a lot of quality control. So you can imagine that if you're going to deliver something into a human, you want to make sure that the quality is super high and everything's well understood. So that's why this is so long. We're also going to be performing toxicity studies in non-human primates. And this again is for safety. So before you go into human, you want to ensure that there's no safety concerns. And the IND is what you submit to the FDA before starting clinical trials. So we can expect to submit an IND in around 2.5 years. We're then going to be pushing additional indications forward. As I mentioned, these have not been chosen, but very likely in the eye and the CNS, in the brain. And so some of these will be pushed also forward into IND, and then 2 more into proof of concept. These are additional advisors and mentors. So Dr. Tony Yag, he was one of our collaborators at UCSD. Actually, I learned a lot of what I needed to learn for pain through him. He's been working in the pain space for over 40 years, and he really understands animal models and pain. Dr. Stephen Waxman, he's a NaV1.7 pioneer and expert. He's discovered a lot of novel mutations in NaV1.7. He's also run clinical trials with Pfizer for erythromelalgia. I couldn't have done this without business advisors. So sometimes I get the question, as a scientist, how do you understand the business, or, what are you doing? Like going from PhD to CEO, like what's that, So I really do think that surrounding yourself with the right people and advisors that understand is super important. So Jeff has helped me with everything from budgeting to hiring to talking to investors to really understanding the process. He's been a CEO of gene therapy companies, so he really understands how to start from the exciting science to actually how to push it towards the clinic. We have also a really great advisor that we met through UCSD, Brady School of Business, and he's been also CEO of multiple gene therapy companies. Mark Wallace is a medical advisor. He's run clinical trials for pain with multiple different drugs, so he understands we can move something forward. Dimas Jimenez is a financial advisor. So you can imagine when you have so much money, you have to have someone that has a lot of idea and control, and understands exactly how to distribute the money and how to control for that. And then very important, obviously, our corporate and IP lawyers. So our corporate lawyer, Randy, he's helped us for everything from starting the documentation to start the company, also to think about strategy when you talk to investors and everything. So he's also been an advisor. And then Matt is our IP lawyer. So one thing that's important is any kind of company, specifically in biotech or tech, is protecting your IP. And so he's really helped us with the strategy there as well. And so just some conclusion, I mentioned have gotten a lot of support from the NIH. We also have collaborations with the branches of NIH called NCATS. We've been given an award, a Mitchell Max Award for Excellence in Pain Research. We've licensed multiple patents. We've had our ITF meeting with the EMA. The EMA is the equivalent of the FDA in Europe. So we had our initial meeting with the EMA. We were featured by the NIH director, Dr. Francis Collins, and we've always been focused on science and publishing high impact proof of concepts as well. So thank you so much for your time, and I'd love to take any questions you might have.
I believe there are 2 so far. So the first one is, how did you know that you wanted to bring your research and idea to market, knowing other businesses kind of do this type of therapy already?
Sure. I honestly think that for me, it started more scientific, excitement, and then I learned business later. So when I was working during my PhD, I thought this made a lot of sense. Like taking CRISPR and repressing this gene in mice and then pushing that into the clinic, it just made a lot of sense to me. After that, I started learning about competition. And the exciting aspect to it was that what I think we're developing is better than what others are developing. Mainly because of the issues I told you earlier. One is the small molecules with NaV1.7 have had lack of specificity. So they'll bind into the heart and have off-target effects. But there's also other gene therapies that target the RNA instead of the DNA. But the issue is that the gene that we're targeting, NaV1.7, is very plastic. What does that mean? Is that the expression of this gene increases with pain. So you can imagine that if you're targeting RNA, which has the amount of molecules change over time, then you're going to have issue with dosage. So how do I know how to bring the research to market? For me, it was when you started seeing exciting data and proof of concept. So really focus on the science and letting the science speak for itself is the most important aspect here. If the science is not speaking, then obviously you can't move it forward. But if it's exciting and it looks promising, then just surround yourself with a great team to push that along.
Perfect. Thank you. Our next one is, what was your pitching experience like when asking for funding for your business?
It's definitely been a process. So, I started this in 2018, and if I showed you my first deck, it would probably make you laugh. Because again, there's a lot of learning that's happened along the road, and I think that what's been really helpful is having grants and really thinking about what we want to do, what's our vision before we start asking for money. So really understanding what's your strategy before you even ask for money. When we first started, though, we did start with pitch competitions. So UCSD and San Diego has some pitch competitions, other students that were also pitching their own ideas. And I know that across the US, there's other competitions. I really would recommend that because it makes you think about how do you tell your story, how do you show the vision. And slowly but surely, I've gotten better over time. Like I mentioned, it was pretty wonky in the beginning, but slowly with a lot of advice, feedback, even when you start getting questions from investors, you start thinking, okay, I keep getting the same question over and over. I should probably put it in my pitch deck. It's something I probably should start integrating into the pitch deck. It's also that kind of experience and feedback that you start getting, and also surround yourself and ask for help. I mean, there's a lot of great mentors out there for business that can help you with your first deck.
All right, so the next one would be, you have been able to raise a large amount of grant funding and are in the process of doing a Series A capital round. What do you think has enabled you to do so well in raising capital, and have you faced any barriers?
Sure. So first, I would say that it's very, what we're focused on first with pain is very timely in the US. There's an opioid crisis, so there's a lot of support from the NIH for that specific disease. So I would say that, it's a combination of exciting technology and uniqueness in the technology for this specific target, but also that right now the NIH is focused a lot on this disorder. And that can change over time. Maybe in a few years, they're going to be focused on other aspects, for example, traumatic brain injury, or Alzheimer's might be the focus. So it's part of that as well. I also would say having a co-founder is super essential. So my co-founder Fernando and I, we're on the ground running, and he writes one grant, I write one grant. We help each other. So it's also about, I think having a co-founder, in my opinion, is one of the most important aspects of being able to do that. In terms of the Series A capital, I can say that that's been a progress in a process. When I first started asking for capital, I was asking for $10 million. And I would receive some term sheets, meaning like some investors wanted to invest, but the terms were not great. So we actually received 7 term sheets and we turned down 6. So it's definitely been a process of like, okay, these people want to take the whole company away, or these people want to move me into a different position, but I want to be CEO. So I can't take that. So again, it's been a process, and it's really finding people that align with your vision and investors that align with you, or else you're going to have a really hard time as soon as you get the money. So yeah, we have faced barriers. Like I mentioned, one of them specifically, they want to put in the gray hair CEO in your spot. So it's really standing in your ground and saying, no, I'm going to be CEO, and I've been successful so far. And I'll surround myself with the right team to make sure that we're successful in the future.
Perfect, thank you. We have one more. Knowing what you know now, is there anything you would have done differently when you were first starting out?
That's a really good question. I think sometimes things happen for, I mean, it's kind of sound like, like something you read a Hallmark card, but things do happen for a reason, in the sense that I feel like if we would have gotten the first money that, so some of these term sheets, we wouldn't have merged the company. We wouldn't have this exciting platform. I think one thing I would change is understanding a little bit more of what investors are interested in, right? So understanding they'd like to invest in platform. They like to invest in, right now, for example, delivery for gene therapies. They like certain diseases more than others. Actually, there's a lot of investors that are scared of pain because of the placebo effects in the clinical trials. And so that's been a difficulty for us. And finding investors that are okay with that, it's been challenging. I wouldn't change anything though. It's just more understanding and trying to find the right investors before even speaking to them, and understanding what they want before speaking to them. So it's always understanding your audience, I guess, and understanding what are they looking for, and if there's alignment there.
We had one more come in. Has it been challenging to put together your team? What have been your main considerations when building your team?
Yeah, I think obviously you can't do anything without your team. It's the most important thing. Even before team and science go hand in hand. I think that the challenge is when you're starting out as a company, the amount of money you have is not very high. And so maybe the salaries that you can pay an employee are not going to be what they can get paid in the market or with other pharma companies. That's been a challenge, right? And you also want to pay people what they deserve. But we have in the early stages, we were able to find some people that were really excited about the science and very motivated by that. And obviously their salaries have increased now that we have more capital. In terms of the main considerations, some feedback that I've gotten is don't hire people that make sure that some part of your team comes from industry, not just academia. Meaning, great, if someone's an academic, then goes to industry and then goes into your company, that's great. But if you only hire PhDs, you're going to be in an academic lab. And really, when you're in industry, there's quality control that you don't have in lab, and quality assurance and all these other aspects you don't think about when you're in research. And the other aspect is making sure that you build the right culture. So making sure that people are happy and excited and treating everyone well, and everyone, doesn't matter up or down, that everyone's a nice person. I think it's a huge aspect. You want to make sure people are comfortable with each other and happy.
We did have one more come in. Did you have any challenges being a female entrepreneur?
Yeah, I think sometimes we don't realize as females that there is some bias. And I did have there's an accelerator at UCSD called My Startup XX, and it brought together all female founders. And something that we learned in that class is that males get more money than female founders. So it's more likely that they'll get funded versus females. It's more likely that investors would put a negative tone towards you versus so for example, they'll say, oh, do you think this is going to be a challenge moving ahead? Versus with the male, it might be more positive, in a positive light. And so there have been some challenges. I have gotten the question of, oh, this is the first startup you do, or you seem young for what you're doing. But my co-founder always points those out to me, and it's just about understanding that they're there and kind of just being positive and overcoming, and always being forward thinking when you answer your questions, right? And being very confident, making sure you're confident in where you are.
Cool. And we had one more. Coming from the deep science side, how hard was it transforming yourself into a strong business person? What do you wish you'd known when you started?
I would say that I'm still learning on the business side, to be honest. I think I'm really excited about business though, because it really allows you to expand on your vision and think about how do you push multiple products forward. In terms of how I become better at business is, again, having a lot of conversations with people that have been there and listening to your mentors and founders, going to conferences that are business oriented in gene therapy, for example. In general, I would say that one of the hires that we will be bringing into the company is either a VP of business development or a chief business officer, because I do think there are some nuances in business that one might not have, or one doesn't have time for. A lot of it could be the Rolodex, right? These people have a lot of contacts. Another is they understand structure of a business. Okay, if you have a partnership, how are you going to collaborate? Who's going to take on what role? What's the amount of money coming in? What are the milestone payments? IP. So all these things might require someone that has more expertise. What do I wish I would have known before starting? That's a good question. I think maybe if I would have taken more business classes, it would have been helpful. I started doing that my last year when I wanted to do it, but maybe starting earlier, right? Even if you don't know if you want to go into business, I think any career, doesn't matter if you're in science or not, you can definitely take advantage of business classes.
I think those are all the questions we have for today. That was a great turnout.
Great. Thank you so much, both of you. Always excited to connect with students, and please go ahead and share my email, and I'd be happy to answer any other questions they might have. Perfect. And to the students out there, hopefully you can take a entrepreneur role, I can say it's been really exciting. You are learning every day. And I think you're learning really in a really fast pace. And it's not something I would have done if I would have gone to industry. It's just, there's regulatory, there's IP, there's finance, there's team building, all these things coming at you at once, which might be overwhelming, but it's also really rewarding.
Well, we want to thank you so much for having you here today with us. We will be putting this webinar on our website. So if you want to let anyone know that you were here, you can always redirect them back to us over here. And hopefully we can share this video out with as many people as possible.
Great. Thank you so much for the invite. And
Thanks
looking forward to it.
Thanks for coming, Anna.
Thank you. Take care.
Bye.
Bye.